My name is Kirshni Hiraman. I am from the HPP lab, which is HIV Pathogenesis program, and we are based at the University of KwaZulu-Natal. HIV is the basic focus of HPP, and we do our research from patient samples that we have, which consists of blood and body fluids. The blood and body fluids are then used in the core lab and separated out for our downstream assays. The downstream assays are flow cytometry, as well as PCR and even sequencing. So welcome to the HPP lab.
Hi, I'm Kireshni and I'm a postdoc at HPP. So this is specimen reception. This is where we receive the samples from our clinics, and this is where they extract the PBMCs, or the mononuclear cells, from the whole blood, and they freeze it down in liquid nitrogen. So now we're going to go through to the immunology lab. So this is our immunology lab. So the first thing that happens is we get the samples that are cryopreserved. So I have a sample here, so let me just get my PPE on. So when we work in this lab, we have to wear all PPE, which is our lab coat, our gloves, and a face shield. Okay, so these are the cells that are still on dry ice. So we're going to thaw them in a water bath that's set at 37 degrees Celsius. So once the sample is thawed, you can transfer the sample to media that contains antibiotics and nutrients for the cells to grow. So the freezing media contains some DMSO, so we first want to wash that away. So we'll centrifuge in the media for 8 minutes. Okay, so after 8 minutes, we're going to decant the supernatant. So we'll have a pellet of cells, and then I'll count the cells. So we're just going to add a little bit of media so we can count the cells. I'm going to count in trypan blue, so it's a blue dye. And what will happen is that any cells that have cell membranes that are disrupted will take the dye in, and so we'll know these are cells that are either dead or on their way to dying. The viable cells will be clear. So I'm going to add some of the cell suspension into the trypan blue. A quick mix. and then load it onto our slide. And then we'll be able to view it under the microscope. So under the microscope, I'm able to see the cells that are viable, so they are intact. They'll be clear, and the dead cells would have taken up the dye, and they'll be blue. So now that we've looked at the cells under the microscope, we know how many cells there are. And now we'll be able to suspend our cells at a certain concentration for staining for flow cytometry. Okay, so I'm going to add 1,000,000 cells. So I've made up the cells to a concentration of 10 million per mole, so that I plate out 1,000,000 cells per well. And then we can spin this down for 6 minutes. So we'll have a nice cell pellet to be able to stain with our antibodies. So we're going to set up our flow cytometry experiment. We're going to stain the cells with monoclonal antibodies that have fluorescently labeled tandems. We're going to stain for CD8 cells and look at their activation markers using CD38 and HLA-DR. So we'll make up the master mix for the antibodies. So I'm going to add 2 microliters of each antibody. So I have CD8, CD3, CD38, which is our activation marker, CD4, and HLA-DR. So what will happen is, once these cells have spun down, I'll decant the supernatant and add the master mix. So the antibodies will bind to the receptor of interest. So for CD38, it will only bind to the CD38 proteins on the cell surface. And each one has a differently labeled fluorescent tandem, so we'll be able to distinguish it on the flow cytometer, the different markers from each other. So these here are our compensation beads and rainbow beads. So they just to help us calibrate the machine and to make sure that everything is working fine from day to day. So this is our plate. You can see the cells underneath. Can you see them?
See?
So again, we're going to decant the supernatant and then add the fixative. And transfer this to a cluster tube. Now our sample is ready to look in the flow cytometer. This is our flow cytometry room. We have 3 cytometers, the caliber, the Fortessa, but today we'll be working on the LSR too. Okay, so this is the flow cytometer. Inside are 3 lasers. So each laser will emit a certain wavelength. So the antibodies that we stained the cells with will be excited by one of these lasers. And so the wavelength will then, it'll be excited at a certain wavelength and then emit another wavelength, right? And then that wavelength will be captured in filters and then will be converted to data that we can see on the computer screen. So I'm just going to log on. So we open the experiment. So with each experiment, to make sure that it's standardized, like across different days, we'll run a control to make sure that the machine is still working the same way that it was yesterday or a week ago when we ran the experiment the last time. So here, we insert the tube. So what will happen is the sample will run through this metal rod up into the machine and past the lasers. So if you run the sample, these are the different fluorochromes that we are using in our panel today. And you can see that the peaks lie in the gates. So everything is working the way it was the last time. So I can now record this. So now we can have a look at our sample that we stained. So my sample is running through the machine now, and the data is being collected and transformed into a visual on the computer. So in this first plot, we're gating lymphocytes, so all of our mononuclear cells. And then from this gate, we look at single cells, and then our viable cells. So if you look in this gauge here, it's not very clear with this color. These are our dead cells that we saw under the microscope. These are viable cells. So we'll only look at the viable population to look at CD3 positive cells. So these would be the ones that didn't stain for the CD3 antibody and those that did. So these are CD3 positive cells. And from here we look at all our T cells. We can look at CD4 T cells and CD8 T cells, and then look at their activation status. So the activation would be your CD38 and HLA-DR marker, which is double positive if they are activated. So you can see our CD4 T cells, they're not very activated, but here we have, if you look at this, 4.8% of our CD8 T cells are activated. Okay, so now we've looked at the activation in these two cell populations, but this is just one patient. So depending on the research question that we are trying to answer, we can either look at patients that are HIV negative versus HIV positive, or we could look at patients that are not on treatment versus patients that are on treatment. And so we will have to do quite a few patient samples for each group, and then we would analyze the results from that on a different software. And then from here, after analyzing the results, those results hopefully will make it to publication.
Hi everyone, my name is Zagiti and I'm a PhD student here at the HIV Pathogenesis program. So today I'm going to be taking you on a tour through our molecular lab, and this is where we do all our molecular work. So now we're inside the lab. In this lab, we have a particular workflow where we work from clean rooms to dirty rooms. Dirty refers to the presence of amplicons, but don't worry, you'll get to understand that as we tour the lab. The first room that we're going to start with is one of the clean rooms, which is the extraction lab. So we're now at the extraction lab, and this is where we extract RNA and DNA from different human samples, whether it's blood, tissue, or plasma. So let's go inside. Hi, Nelly. Can you tell us a little bit about what you're doing?
So hi everyone, as Zagita has mentioned, that my name is Nelly, and today I'll be taking you through the extraction room. So in this room we extract both RNA and DNA from blood samples. So we use plasma and we use different kits. So in this room we have different kits from different companies, and what we'll be showing today is the QIAGEN viral extraction kits that we use to extract viral RNA from plasma. So what is very important in this lab is that you have to cover your face to protect your eyes from any splash that might happen during experiment. You also have to double glove. We use both nitrile and latex gloves. And so today I'll be starting my experiment. So when we do our experiment, we work in a fume hood, and we do all our experiments inside the hood. So this is good for our protection as well, and it also regulates the airflow inside the hood. Today we'll be working with plasma. Plasma is extracted from blood samples. So what happens is that when you get a blood samples, we spin it using a centrifuger, which is this machine over here, at high speed. And so all the red blood cells, the white blood cells, and the platelets are spun down to the bottom of your tube, and then all the liquid that is left on top is your plasma, and that's what we use to extract our viral RNA. Okay, so the viral RNA has three very important steps. The first one being the lysis, where we release the DNA inside the virus. And with this one, we use a viral lysis buffer. We'll then precipitate our DNA and wash it with wash buffers. These normally contain mostly ethanol, and then we will then elute it and in our elution buffer, and that will be used for downstream purposes. So right here we have our tips and our pipette tips. We use these tips to add our liquids or our reagents to our tubes. So we'll be using these 2 ml tubes, And this is where we'll be adding all our reagents using our pipette tips. So at first, when we start with the viral extraction, we will start by lysing our plasma, where we'll be adding all our reagents into this tube, along with a carrier RNA, which is one of the enzymes that helps with the lysing buffer. We will then spin our plasma or the mix-up into the centrifuge. We will then add it into our spin column. So once the lysis has happened, all the nucleic acid will be released into the solution, and we're going to use this column to trap the DNA while the rest of the cell debris run through the column. We will then wash our viral RNA with these two wash buffers, which mainly contain ethanol, and then we'll elute it using this elution buffer, and then we can do other downstream processes. Yeah.
So now we've seen an extraction lab. Now we're going to move on to another clean room, which is our master mix lab. So this is the last clean room that you can go to, and like I mentioned before, once you're dirty, you cannot come back to the clean rooms. A major focus in our molecular lab is polymerase chain reaction, or PCR. So this works like a photocopy machine where we make lots of copies of our particular gene of interest, which we're looking at in the HIV genome. So let's see what happens in the master mix lab.
So in the master mix room, this is where we make our master mix that we need to make copies of our PCR. So these are some of the reagents that we need. First of all, we're going to need water. This is not just any water, this is nucleotide-free water, which is free of any RNAs and DNAs. We're going to need our buffer. We use buffer to regulate the pH inside the reagent to be making our PCR. We also use our magnesium chloride, which is salt that makes a suitable environment for the reagents. So these are our primers, and we use to amplify our gene, and these actually start to the beginning of our gene of interest. And we also have our nucleotides, which we add to actually extend our DNA. And the last one is our enzyme, which is our catalyst. So once we've made our master mix, we then aliquot it into our 96 well plate. So depending on how many samples that you're working with, you can either work with a big plate, which can take up to 96 samples, or you use small strips, which can take up to 8 samples. Once you're done adding your master mix, we then use this adhesive film to seal off the plates, and then we'll take it to the amplification room.
So now we've come from the master mix lab where we've added all our reagents. You can kind of think of this like baking a cake. So we've added all the ingredients we need to amplify our gene of interest, which is done in the amplification lab. So inside this lab, we have thermal cyclers, and these are the machines that we use to basically bake our cake. So let's go and see what they look like. So these are our thermal cyclers, which we use to perform our PCR. This particular one is called the PCR Max, and we use this to make many copies of our particular gene of interest, which we're going to use in our downstream processes. So in this process of amplifying our particular gene of interest, there are 3 major steps. What this machine does is it controls the different temperatures which are needed to firstly denature the DNA. So this is to open the strand of DNA that we want to amplify our gene of interest. The second step is the annealing step, where we use our primers, which Nelly mentioned that she added in the master mix room, to amplify our gene of interest. So what these primers do is once the DNA is open, and we have 2 strands. It scans along this DNA to see where it must bind to amplify, and that's make copies of our gene of interest. Then finally, we have an extension process, and this is where the DNTPs, which Nelly mentioned as well, are little nucleotide, little pieces of DNA that add on and make the strand of DNA which we're interested in. So following coming from the amplification room, we want to go and view our results. So whether we've been able to successfully amplify our gene of interest. So this we do in the electrophoresis lab. So here we are now at the electrophoresis lab, and this is where we actually run our gel using gel electrophoresis. What this does is that it separates different pieces of DNA into their sizes. So because we know what size the particular gene of interest we're looking for, we'll be able to view it on a gel, which is what you'll see just now, using our gel electrophoresis machine. So let's go inside. So Dottie is here viewing her results. Hi, Dottie.
Hi. I want to show all of you what happens in this electrophoresis lab. So in the electrophoresis lab, once you're finished with all your PCRs, this is where you will now view your results. This is the moment of truth. So here you'll have to make your buffer and make your gel. So basically what we do is take the buffer, you measure it, depending on the amount that you need and the tank that you're running. So this is the flask that we use, that is the buffer that we add. Then we add the electrophoresis tablets. So once you do that, you leave this to dissolve. And I have one that has dissolved already. So once it dissolves and dissolves perfectly, you boil it. So, and in here I have already boiled gel. So this is how your gel looks like before you load it. Of course, it's hot, you will have to cool it down. So in the lab, because we are water conservatives, we do it by you put some water in your beaker and you rest your gel, you let it to cool down. Once it has cooled down, I'll show you a sample that you pour. But while this is cooling down, so you'll just rock it up and down, let it cool down perfectly, so that it won't crack the tanks. So basically what I have here is a loaded tank. Then you'll add your gel. Gently, you pour it gently. Yes, with all the wells. Quite gently. And that basically it. We put everything back to the sink that will be cleaned up later. So here I'm showing you a full loaded tank that is about that has cooled down and has set to form the gel and is about to be loaded. Here I have one of my colleagues who's been loading a gel. And basically what it looks like once you've added your PCR products, because I'm sure through the PCR, these are the tubes that you had. You won't see anything here. It will be just your master mix and your amplified products. Basically, after you do that, you'll have you'll load your template using a loading die, and it will look blue in the gel. If you can check through that. So that is already running using the power tank. And basically that's it. Once the gel has run, maybe earlier on you had the alarm going through. So once you've run your gel, you'll bring it here in the ChemiDoc, and clearly this is what you'll place your gel there, and this is the image you'll be able to see. Yeah, so this shows you what this is the moment of truth, that actually what you've been doing in the lab has actually shown some results. These are happy bands, and this is a happy scientist, basically. Then, so going forward from here, once you've had your PCR products, you've done the gel loading, and you've viewed your gel, then the next step from here is to do sequencing. And sequencing basically is to see what kind of products do I have. Are they really the target products that I was looking at? And what are they telling me? So for you to get that information, you have to do the process of sequencing. Then when we do sequencing via another department, we process our plates, then we get results. This is how your result will look like. Basically what it shows in this output, when you see a red, it shows that that was a poor, that is not the sample, the sequences were not so good when it shows a red. When it shows a yellow, they were medium, but when it shows a blue, those are perfect sequences, and clearly you can see more of this because of the beautiful gel we viewed earlier. These are more blue than red, clearly showing that the sequencing worked. For your sequences, then you'll be able now to have to run it in a sequencer file, which I'll go through basically for now. So what you'll get once you get that read, you'll get an overview of your sequences. From the overview of your sequences, you can go and check in. Here it just looks like the overview. Yes, it covered the whole beginning and the end of the sequence. Then you go to the bases. These are the bases. I'm sure by now you've already been informed about the ATG. Those are the nucleotides. And from there you are able also to see the electropherogram, the peaks, and what the color symbolizes. Like A is red, C is blue, and T is green in this snapshot that I'm showing here. So once you've done that, you are able to analyze your data further and further, and this to make it publication ready. But basically, like what I'm doing personally in my project is to see that sequences are compared from the patient, and what you generate in the lab are actually similar or they are comparable. And in this image here, I have a picture of a tree, a phylogenetic tree that shows very, very many sequences from 487 patients. There's also sequences that will just show partnerships, whether it's from mother and child, and whether it's from husband and wife, viruses extracted from these particular people.
So Darty's just shown us everything that happens in the electrophoresis lab. So the last lab that we're going to take a look at up here in our molecular lab is called the cloning lab. And no, I'm not talking about cloning people. So in this lab, what we usually do is take our particular gene of interest, which we have amplified, as I've shown you in the amplification room, and then checked if we actually have this gene of interest. So we take the small DNA fragment and we clone it into something called a vector. Our vectors are usually plasmids, which are carriers of DNA. So they carry this little piece of DNA which we've amplified and is of interest to us. So once we have it in the plasmid, we can use for even further downstream processes, such as looking at expression of the particular gene, or looking at or creating HIV replicant competent viruses, which we do downstairs in our biosafety level 2 lab.